Hello everybody and welcome back to another RevWise video. Today we're going to be doing another IGCSE Edexcel double award paper. This is a 1BR paper, so it's the same as 1B but just a regional version of the paper. This will be very good practice, and I know everyone has their exams soon, so best of luck for your exams, and I don't want to waste any time because I know your exams are very soon, so I'm getting this done on short notice. So let's go straight into the questions. Question 1. The diagram shows a part of a food web with the desert community. How many organisms are secondary consumers? So let's have a look. We know that the first thing in the food web is going to be the producer, right? And we know the second thing, the arrow that goes from the producer, is going to be the primary consumer. So these are primary consumers. So what are we looking for? We're looking for the arrow that goes from the primary consumer to something else. So we can see that it goes from the scarab beetle to the monitor lizard. So that is a secondary consumer. It also goes from the desert rat to the monitor lizard. But that is the same organism, so there's still only one secondary consumer. But we can also see it goes from the desert rat to the fennec fox, so that makes it two secondary consumers. So even though there's two arrows going here, there's only one monitor lizard, right? So you don't count it as three. The answer is 2, which is A, and I hope you can understand that. Okay, now I want you to draw the longest food chain in the food web. So drawing food chain is pretty simple. It's just going in one direction, going right. And you just find the longest chain. So you look where the most arrows lead from. So let's have a look. We can see acacia plant. Acacia plant goes to scarab beetle. Goes to monitor lizard. Goes to hawk. So this is 1, 2, 3 arrows. Okay, let's look for something longer. Okay, acacia plant, 1 to desert rat. Then to monitor lizard, another 1. So that's 2. And then monitor lizard to hawk. That's 3. Okay. Now let's look at another one. Acacia plant to desert rat. 1, desert rat to fennec fox, 2, desert fennec fox to monitor lizard, 3, and monitor lizard to hawk. That's 4. And we can see that anything else will be shorter. So we have a 4 arrow food chain. So now we just have to write that. So we just do acacia plant, then goes to desert rat, which goes to fennec fox, which goes to monitor lizard, which then finishes at hawk. And there we go. And that's how you draw the food chain, so just be ready. You just have to look for the one that reaches the apex predator, and you want to see which one takes the most arrows to reach it. So basically that's all you have to do. Okay, now another common question. I saw it as soon as I read it, I know exactly what to answer. So explain why most of the energy of producer is not transferred to the hawk. Well, it's a very common question, and basically, if I'd say, I think this is probably a question like this might come up on your paper because it's very, very, very common, and it's just talking about why energy is lost in the food chain, and it wants you to apply to the situation. So you just can say the general answer. So one easy answer to say is that energy is lost due to excretion slash metabolic waste, essentially. So this is one thing. So remember, excretion is like the removal of waste products on metabolism. Don't get it confused with egestion, which is to do with feces. Egestion is to do with feces. Excretion is to do with waste products on metabolism, right? So urine is to do with excretion. So you can say energy is lost due to excretion. We can also say that energy is lost due to indigestible parts of the producers. Right? Producers or anything, you can just say indigestible parts, because you're just saying that some parts aren't able to be digested, so their energy isn't carried over. Energy lost from heat loss during respiration. So remember, if you mention heat loss, it's always good to also say respiration, because that's the actual process. And finally, energy lost during movement. Oh, another key one you can say is energy lost from some parts of organism not being consumed. And if it's confusing, just think of bones. I mean, most animals do not eat the bones of their organisms, so that's energy is some energy might be lost there. There might be some parts that animals just don't like eating. Then we're just losing energy along the food chain, along the food web. So yeah. Okay, this photograph shows a fennec fox. Very cute. Fennec fox live in the Sahara Desert, which is very hot. They have large ears and a thin body. Explain how the body shape of a fennec fox has evolved by natural selection. Okay, so natural selection, very simple formula we use for answers to natural selection. First, we have to say when natural selection, the first mark usually, nearly always, is a mutation occurred in the genome of a fennec fox. So somewhere along the line, there's a genetic variation or a mutation that occurred, and which caused a fennec fox to have a large ear surface area and a thin body. Okay? So due to this, right, this allowed the fennec fox to stay cool, right? Because we know that larger surface area, thin skin, because they're in the desert, more they have larger surface area to lose their heat, right? To stay cool, as they have a higher SA to lose heat. Okay, so then this this mutated fennec fox is able to survive more, allowing for production of offspring with the same body shape. Okay, so they survive more. It's promoting them breeding, passing on their genes. So this passes on their alleles for ear shape / thin skin or whatever, thin body, to the next generation. And that's basically all it is. So think of that formula, so mutation, and then mutation enables them to have some sort of characteristic that allows them to survive more, and because they can survive more, they're allowed to reproduce more and pass on their alleles / genes to the next generation. Okay, question 2a, the eye. Okay, eye is interesting. I found it quite hard when I was doing GCSEs, and not hard, but it was quite complex compared to other units. So let's see, structure of an eye, which is a structure labeled X. Okay, so X, we can see it's pointing to not the outermost, it's not pointing to the outermost layer of the eye, it's the thicker layer below that, and we know that should be the cornea. If I can tick the box, there we go. Okay, yeah, so we should know that. When looking at a close object, which row of the table shows the state of ciliary muscles and suspensory ligaments? Very important unit. Okay, let's have a look. Process of elimination. So ciliary area is contracted, suspensory ligaments loose, contracted and tight, relaxed, loose, relaxed, tight. Okay, so when it gives you like a MCQ about these suspensory ligaments or muscles, they usually you can immediately cancel out some of them because you know it can't be B or D. Why? Because tight is not the terminology we use. We call it contracted or loose. That's just what we say, right? So we know it's going to be A or C by process of deduction. And if you know, you should know that ciliary muscles are contracted when we have an object that's close to us, and the suspensory ligaments, they loosen, they relax. So the answer should be A. And yeah, just remember that for close, the way I remember it is close ciliary contract. So triple C, right? And if you only need to remember one of them because you know the other one's going to be opposite, right? So if you remember close ciliary contract, you know that when it's close, when it's far, ciliary relax, and in turn, you know that suspensory when close is loose, and then when suspensory when far is contracted. So yeah, there we go. Okay, I explained that structure Y controls the light entering the eye when someone walks into a dark room. Okay, let's look. So structure Y, okay, and so structure Y, we know what it is, iris, and it controls the size of the pupils, and it wants us to talk about how it. Okay, this is a nice question. So when we enter a dark room, remember that we want more light if it's dark, right? So we want our pupils to become larger. So pupils dilate. That's the right word you should be using. Pupils dilate, right? And then it wants us to explain how structure Y controls it. So it wants us to talk about the individual muscles that does it. So how do we say it? So when we do it, circular muscles, so remember there's the two muscles, circular muscles and radial muscles. So circular muscles contract. Radial muscles relax. Wrong way around. Circular muscles relax. Radial muscles contract. Okay? So remember that when you enter a dark room, the radial muscles are the ones that are gonna to make your eyes bigger. The radial muscle is the one that's gonna make your eyes get larger, and then your circular muscles are the ones that are gonna help your eye contract. So in the dark room, your circular muscles relax, radial muscles contract, and it's quite easy to actually imagine because you can think of it like how a circular muscle is um, kind of is here. So where this is the pupil, this is the pupil. A circular muscle kind of is in this area. So this is where the circular muscle acts, and it kind of pushes it in. Whereas the radial muscles, they're over here. These are radial muscles. So these are moving. They can expand the pupil by moving in the opposite direction. But you don't need to know that. You just need to know this. Just try to remember. It's a simple thing to remember. In a dark room, circular muscles relax, radial muscles contract. There we go. Okay. Okay, now B. Multiple sclerosis is a condition that can slow down the speed at which electrical impulses travel along neurons. Time taken for a blink reflex to occur can help diagnose a person as multiple sclerosis. Okay, this is the blink reflex. There's one to the eye, and time is recorded. Okay, this is the pathway right here. The speed the impulse moves along the reflex it consists of 3 neurons. This person, multiple 70 centimeters. Okay, time taken for the blink reflex to occur in a person with multiple sclerosis is 0.005 seconds. Okay. So without multiple sclerosis is 77. With this time, total length of the neurons in the reflex arc for the person with multiple sclerosis is 25 centimeters. Calculate the difference between the speed of impulse for the person with multiple sclerosis and the person without multiple sclerosis. Okay, let's see. So we know that the speed of the first person is first without multiple sclerosis is 77 meters per second, and then we need to get the person with multiple sclerosis. So we have the speed is in meters per second for the first one. We can see 77 meters per second, and so we're going to just convert this one to meters per second as well. So we already have the time in seconds. So speed equals d over t. So speed equals distance. So we need to get our distance in meters. So 25 centimeters, 0.25 meters. Divided by our time of 0.005 seconds. So that gives us 0.25 divided by 0.005. And that should give us something around 50 meters per second. And now it wants us the difference, so 77 minus 50 equals to 27. So that's our final answer for this question, 27 meters per second. Okay. The speed of an impulse along the axon of the motor neuron for someone without multiple sclerosis is 120 meters per second. So just why the speed of the impulse calculated along all 3 neurons is less than the speed of the impulse along only the motor neurons. The speed. Okay. So it's asking why in the motor neuron the speed is like higher than all 3 neurons combined, and the reason is across all 3 neurons there are more synapses which travel by chemical diffusion of neurotransmitters. Okay, and then I would do an explanation, but the question says suggests. So remember, you might time actually isn't that big of a concern usually, but if you are usually find time concerns, make sure you read the command terms so you include only what's necessary. So it suggests. So we're just going to suggest why, and the reason is because there's more synapses, and we just explain what the synapses happen. Okay. So we have a yeast cell. Okay. And it wants us to label where. Okay. Where does aerobic respiration occur? Oh, we know it occurs in the mitochondrion. Very simple. What substance is the yeast cell wall made of? Well, we should know that it's not cellulose because that's plants. It's not glycogen because that's how fat is stored in animals, and you know it's not starch. And so the substance of the cell wall of a yeast is actually chitin. Okay, which of the structures found in the yeast cell wall would also be present? Found in the yeast cell would also be present in a prokaryotic cell. Okay, so the yeast cell would also. Okay. The answer is not mitochondrion, because we know that doesn't exist in there. We know for sure there's no nucleus. and unless there's very, very specific cases, there's no vacuole, so we know that it's cell membrane. And it makes sense. I mean, you can't really have a cell without a membrane most of the time. Okay. Yeast also used to make bread, and this is the procedure that the student used to investigate the temperature and height of bread. Okay, table shows results. Calculate the percentage increase in the height of the dough after 2 hours at a temperature of percentage increase of 35 degrees. Okay, so it wants us to calculate the percentage increase for the 35 degrees, which is 45 after 2 hours and 25 after start. So we go into the formula final minus initial over initial to get the it. So we get final, which is 45, minus initial, which is 25, all over the initial, which is 25, and then we get an answer of 0.8, and then we times that by 100 to get a percentage of 80% increase. Nicely done. Okay, explain why the yeast causes the bread to increase the height. Okay, so we know that for yeast, when yeasts respire anaerobically, when yeast respires, it doesn't even have to be when we can say anaerobically, it's not 100% necessary, because either way we know that when yeast respires, carbon dioxide is produced, and this forms bubbles inside the bread dough, which inflates the dough. Okay, pretty simple one. Explain why the dough rises to a different height at 25 compared to the height at 35. Okay. So for this one, it wants us to explain why. So we can just explain at one of the temperatures, we can just explain why it's lower or higher. So we can see that at 25, it's a lower height instead of 35. So we can explain it saying at 25, right? There is lower kinetic energy from lower temperature, so the particles move slower. Okay? This means that there are fewer enzyme substrate complexes formed from successful collisions. Excuse my spelling, I'm very sorry. From successful collisions. Okay. Fewer enzyme substrate complexes formed. You can also say that it's further away from the optimum of the reaction, anything like that. For the enzyme optimum, make sure you say enzyme optimum, and then we also have to say that because of this, so it's asking why the dough rises to a different height. So the dough rises to a lower height due to less carbon dioxide gas being produced. Okay, so yeah, that's a simple way of answering it. And another thing, 65. Okay, so we can see at 65, instead of what we'd expect, we'd expected it to increase a lot, because it goes from 40 to 80 and then goes to 8. So when a temperature suddenly drops the rate of reaction, we know that the enzymes are denatured. So at 65 degrees, the enzymes are denatured, so the substrate can't fit into the active site due to I won't use any complex stuff because this is only GCSE, due to a change in shape of active site. So respiration doesn't continue or continues at a slower rate. Okay, question 4. Diagram shows a human alimentary canal. Explain how food passes through the esophagus. Well, esophagus, we all know that lovely word peristalsis. Peristalsis occurs, which is a wave of muscular contractions. Make sure you say contractions, because that's a very relevant point. Okay, so yeah, peristalsis occurs, waves of muscular contractions. Okay, which lipid structure produces bile? Well, we should remember that the liver produces bile and the gallbladder stores bile, right? So it's not going to be D because D is pointing to the gallbladder, and we know it's not going to be A because A is pointing to the stomach. It's not going to be B because B is pointing to the looks like the pancreas. So our answer is C, which is pointing to the liver. Okay, rule of bile digestion. Only 3 marks. Usually this question is 4 marks, but it's a very simple question. Basically, for this question, there's two things you have to say, and you have to explain the use of each thing, right? So the first use of it is bile neutralizes. I guess we could say stomach acid. Stomach acid. So that's the first use because we know bile is alkaline, so it's going to neutralize the stomach acid to set the optimum pH for enzymes in the small intestine or ileum, duodenum, or intestine. Okay, and now we can say the second point is that bile emulsifies lipids, breaking them in to smaller droplets. And what's the use of this? Increases the surface area for enzymes to act on. So yeah, the 2 points are basically bile neutralizes stomach acid and bile emulsifies lipids into small droplets, and then you have to explain each one. The use of it is to set the optimum pH or enzyme in the small intestine and to increase the surface area for enzymes to act on. Simply done.